Let's begin genetics part 2, predicting offspring. If I have 3 girls, am I more likely to have a boy next time? The answer, of course, is no. However, this is a common misconception or misunderstanding about how the numbers work in inheritance. Today, we'll look at a couple of tools to use in addition to a Punnett square to study and predict offspring of specific crosses. A valuable tool in studying inheritance patterns is a pedigree. A pedigree is a family tree that describes the interrelationships of parents and children across generations. Inheritance patterns of particular traits can be traced and described using pedigrees. Let's look at a pedigree more closely. Is the trait shown in this pedigree dominant or recessive? What's the genotype of the firstborn male in generation 2? What's the genotype of the youngest female in generation 2? The trick to reading a pedigree is to look for a place where the parents are identical, but at least one of their children are different from the parents. In this case, look at the parents in generation 3. Both are unshaded, meaning that they do not show the trait in their phenotypes, but one child does show it. This means that the parents must have had the allele in question without showing it in their phenotype, which means this trait must have been recessive or hidden in these 2 parents. Once that is known, the other question can be answered. The chances of producing specific phenotypes can be calculated using the rules of probability. In order for us to determine the chances of an organism inheriting a single gene trait, we need to be able to apply the rules of probability. Mendel's law of segregation and independent assortment reflect these rules of probability. When tossing a coin, the outcome of one toss has no impact on the outcome of the next toss. In the same way, the alleles of one gene segregate into gametes independently of another gene's alleles. The rule of addition can be used to figure out the probability that an F2 plant from a monohybrid cross will be heterozygous rather than homozygous. The multiplication rule, also called the product rule, states that the probability that 2 or more independent events will occur together is the product of their individual probabilities. Probability in an F1 monohybrid cross can be determined using this multiplication rule. Segregation into a heterozygous plant is like flipping a coin. Each gamete has half a chance of carrying the dominant allele and half a chance of carrying the recessive allele. Using the multiplication rule can expedite the process of predicting the probability of producing specific phenotypes in the offspring. Given that, determine the probability of producing green smooth seeds if this is the parental cross. Big G, big S, little s, crossed with big G, big S, big S. What is the probability of producing green, smooth seeds in this cross? Take a moment to pause the video and do your calculations. Here's the solution. Green has 3/4 of probability. Smooth has a 4/4 probability. So 3/4 times 4/4 equal 12/16, or a 3/4 probability of producing green, smooth seeds. The AP Biology formula chart contains the formula to use when applying the rule of addition. The probability of having one girl is 1/2. The probability of having a second girl is 1/2. The probability of having that third girl is 1/2. Each conception is independent of the next. So we would calculate the probability of having 3 girls by multiplying 1/2 times 1/2 times 1/2. That would give us 1/8 chance of having 3 girls. The same would be true of having 3 boys. However, the odds of producing either 3 boys or 3 girls is 1/4 because there are 2 ways to get the result. So the probabilities are added 1/8 plus 1/8, giving us 1/4 chance. Let's look at an example of applying the multiplication and addition rule to predict the outcome involving multiple characters. Here we have a heterozygous cross between big Y, big R, little r. What would be the probability of getting an offspring who has 2 big Y's and 2 big R's? The calculation is shown there. We have a 1/4 probability of getting the 2 big Y's times the 1/4 probability of getting 2 big R's, giving us a 1/16 probability of producing the genotype big Y, big R, big R. If we look at the probability of producing big Y, big R, to get big Y, little y, we have 1/2 probability times the 1/4 probability of producing big R, giving us a 1/8 probability of producing a heterozygous, homozygous, big Y, big R offspring. So we can apply the multiplication and addition rules to predict outcomes of crosses involving multiple characters. A dihybrid or other multi-character cross is equivalent to 2 or more independent monohybrid crosses occurring at the same time. In calculating the chances for various genotypes, each character is considered separately, and then the individual probabilities are multiplied. So you can see how that would be an advantage rather than drawing a giant Punnett square if you simply apply the rules of probability. When considering 2 or more traits, calculating the probability mathematically is much simpler than drawing a Punnett square. I'd like for you to work with a partner to write out how each probability should be calculated. For each of the 4 examples shown below the first one, write out on a sheet of paper how each of those would be calculated, showing your work. Hope your answer looks somewhat like this, and that's certainly easier than drawing the Punnett square that would take to do a trihybrid cross. In Genetics Part 1, we reviewed several vocabulary terms that are important when you're trying to work or solve problems involving probability and genetics. So just to keep the vocabulary fresh, I'd like for you to stop and do the card sort. So take out your set of cards and sort them, matching the term with its definition. So now that you've had a chance to review some vocabulary and also become familiarized with the multiplication rule and the addition rule for probability, I'd like for you to solidify your skill by completing a set of practice problems, and they're available at the link shown on the screen. Once you have your problems completed, you'll submit them to your teacher in the manner they indicate. And that wraps up our session on calculating genetics.